So last time is we tackled about the functions of the parts of a prokaryotic cell. And for today is we will talk about the parts of a eukaryotic cell and its function. First is we have what we call structures for protection. The first one is we have the cell membrane. So cell membrane can be found on both plant and animal cell. So it functions as a barrier to separate the environment and the internal structures of the cell. Also, it regulates the passage of the molecules to and from the cell. So it is composed of two sheets of phospholipid, hence the name phospholipid bilayer. Next one is we have the what we call cell wall. So cell wall is not found on animal cells, but it is found on fungi, bacteria, and also plants. So it is an additional boundary between the cell membrane and the environment for added structural support and protection. So it is only found in plant cells and also in fungi, but also present on prokaryotic cells such as bacteria. And also its composition varies per classification. For example, we have chitin or chitin for fungi and cellulose for plants. Next one is we have the cytoplasm. So don't be confused of this one. So when we talk about the cytoplasm, it includes the what we call cytosol, the organelles, except the nucleus. Okay? Cytoplasm is the jelly-like substance contained within the cell membrane, generally composed of the cell organelles, not including the nucleus, but it includes the cytosol. Now, we have the what we call cytosol. Cytosol is the liquid component of the cytoplasm and is largely composed of water. Next is we have the genetic control organelles. We have here the what we call nucleus. So the nucleus is the storehouse of genetic information known as the DNA or the deoxyribonucleic acid. It functions to prevent damage to the DNA and it directs all the cell activity. It is known also as the control center of the cell. So on the nucleus, it is composed of four parts. We have here the what we call nuclear envelope. So it is a two-membrane structure that encloses the nucleus and separates it from the cytoplasm. Next one is we have the nucleoplasm, the semi-fluid substance inside the nucleus. We also have the what we call nuclear pores, on which these are perforations in the nuclear envelope that regulate the passage of materials. And also we have here the what we call nucleolus, the one that produces the rRNA or the ribosomal ribonucleic acid. We have here the what we call nucleolus. So the nucleolus is the condensed region of the nucleus where the synthesis of the ribosomal ribonucleic acid or the rRNA and ribosomal subunits are centralized. Next is we have the what we call DNA or the deoxyribonucleic acid. It is the blueprint of life. So it is the long chain molecule of inheritance made of segmented portions called genes, which contain the information needed for the cell activities such as protein synthesis and others. The molecule is compacted by proteins called histones, which form a condensed structure called chromatin and condenses further into chromosomes when cell division is occurring. Next one is for the manufacturing, storing, and other functions. We have here the what we call endoplasmic reticulum. So endoplasmic reticulum or the ER is an extensive folded membrane that occupies a large space in the cytoplasm. Membranous tubules and flattened sacs of the ER are called cisternae, while the spaces between them are the cisternal space. We have two kinds of endoplasmic reticulum. We have here the what we call rough ER or the rough endoplasmic reticulum. It is the region closest to the nucleus whose cisternae are flattened sacs and are studded with ribosomes, giving it a rough appearance when viewed from the microscope. Also, rough ER is the one responsible for the transport of proteins using the vesicles. Next one is we have the smooth endoplasmic reticulum. So it is the region adjacent to the rough endoplasmic reticulum where no ribosomes are present and whose cisternae are membranous tubules. Its main functions are mostly involving metabolic processes such as synthesis and transport of lipids. Also, it functions for the metabolism of carbohydrates and detoxification of drugs and poisons. Next one is we have the ribosomes. Ribosomes is the site of protein synthesis. Spherical granules composed of two subunits, the large subunit and small subunit, which is synthesized in the nucleus. So these subunits exit to the cytoplasm and combine to form the ribosomes. Next one is we have the Golgi body or the Golgi apparatus. So these are stacks of cisternae which acts as a processing system that sorts, modifies, and packages products from the endoplasmic reticulum. Protein finish their folding here to become functional, and also enzymes in the Golgi body manufacture and attach carbohydrates that serves as the name tags of the cell. Okay. Other facts about the Golgi body. It is composed of three regions. It is the CIS region or the cis region, the medial region, and the trans region. So for the first one is we have the cis space or the cis region. It is the receiving end of the Golgi body. It is the one responsible for the returning of incorrectly sent proteins back to the endoplasmic reticulum. Next one is we have the dictyosome or the medial phase or the medial region. It is the bulk or main stacks of cisternae. It is the one responsible for processing proteins and lipids and directing them to their destination. And for the last one is we have the trans phase or the trans region. It is the shipping end of the Golgi. It sorts and directs the modified proteins to their destination by budding off secretory vesicles. Next one is we have what they call lysosomes, the suicidal sac of the cell. So it is a free-floating organelle that contain enzymes that dismantle and recycle food particles, captured bacteria, worn-out organelles, and debris. Next one is we have what we call peroxisome. So peroxisome is the one that breaks down fatty acids, and also it serves as a fuel for respiration. So it is structurally identical to lysosomes. However, the enzymes present are from the Golgi body and have much higher concentrations than lysosomes. Its main function is to dispose toxic substances and lipids. Next one is we have what we call mitochondria. It is known as the powerhouse of the cell, and it is the center of respiration of the cell. It gives energy to the cell by means of cellular respiration. And also, it contains oxidative enzymes that converts sugar to ATP or adenosine triphosphate. It consists of their own ribosomes and DNA, which is the basis of the endosymbiotic theory. The endosymbiotic theory or the symbiogenesis is the theory of the origin of eukaryotic cells from prokaryotic organisms. The mitochondria is a membrane-bound organelle. It contains the what we call outer membrane, which is the smooth, continuous boundary that encapsulates all the contents of the mitochondria. Next one is we have the inner membrane. So it has intricately folded structure called cristae, where the enzymes responsible for the cellular respiration are located. The number of cristae is proportional to the energy requirement of the cell. In between these folds is the mitochondrial matrix. Next one is we have what we call plastids. So just like mitochondria, so plastids are the basis for the endosymbiotic theory. So plastids is a group of large organelles only found in plant cells and not in animal cells. It can be divided into three groups. We have what they call chloroplasts, chromoblasts, and leucoplasts. First type of plastid is we have the chloroplasts. These are green-colored plastids due to the green pigments, chlorophyll A and B. Chloroplasts are the site of energy production in plants because this is where photosynthesis occurs. This process makes use of solar energy and transform it into energy-rich molecules. Next one is we have what we call chromoplasts. These are colored plastids rich in pigments such as carotenoids instead of chlorophyll, which give fruits, flowers, and leaves their orange, yellow, and red colors. Chromoplasts can arise from chloroplasts that have lost their chlorophyll, such as the ripening of fruits. Next one is we have what we call leucoplasts. So these are non-colored plastids that do not contain any pigments. They function mainly as storage for nutrients. So starch-filled leucoplasts are called amyloplasts. Those that stores all are called alloplasts, and those that stores protein are called aleuroplasts. Next are the organelles for support, movement, and communication. We have here the what we call cytoskeleton. It is a flexible network of protein threads and fibers that provides mechanical and structural framework of support throughout the cell. Additionally, it helps in directing cell movement, transport, and responses. Next one is the parts of the cytoskeleton. We have here the what we call microtubule. It is the thickest and the longest filament composed of tubulin, and it provides structural support and transportation. We have here the intermediate filament. It is smaller than microtubules and appear to be rope-like. Its function is to provide mechanical support. And lastly, we have the what we call microfilament. It is the thinnest filament composed of actin, which is present in nearly all cell types of eukaryotes. Its function is for elongation and contraction. Next one is we have the centrioles and centrosome. It is a small dense region in the cytoplasm where microtubules are assembled and arranged. Inside the centrosome are two hollow cylinders made of filaments, which are microtubule triplets, which is called the centriole. Centrosome division is crucial in preceding and stimulating the cell division. Next one is we have the what we call vacuoles. Vacuoles functions for the storage of food and water and responsible for the turgor pressure. Next one is we have the cell surface and junctions. Cells are joined to each other by structure called junctions. In plants, the plasma membrane and cytoplasmic fluid extend specialized structures between cells called plasmodesmata to communicate and pass molecules effectively. We have three types of cell junctions. We have the tight, adhesion, and gap. For the tight, it fuses cells together to form an impassable barrier using actin fibers attaching to membrane-anchored protein, forming cell sheets. We have here the what we call adhesion. It connects two neighboring cells together or a cell to the extracellular matrix. And lastly, we have the gap, which is a channel protein that links with the cytoplasm of an adjacent cell to allow exchange of ions and nutrients.